Hello, this is Yashveen. In this video, I'll be solving paper 4 variant 3 October November 2021 Cambridge IGCSE Biology. 1. Enzymes are used in genetic engineering. A. Define the term enzyme. So enzymes are biological catalysts. B. The process of genetic engineering often starts with the steps in figure 1.1. So you've got the two strands of DNA, an enzyme which is going to cut the two strands of DNA. 1. State the sequence of bases on the lower strand of DNA molecule in step 1. So the lower strand of DNA would have bases that are complementary to these, and so those would be C, T, T, A, and G, as G and C are complementary and A and T are complementary bases in a DNA molecule. 2. State the name of the enzyme in step 2 of figure 1.1. So this enzyme is going to cut the two DNA strands, so it is a restriction enzyme. Three. Describe the effect of enzyme 1 on the DNA molecule in step 3. Restriction enzyme will cut the two strands of the DNA molecule in an uneven way, which will produce sticky ends. So these sticky ends are just unpaired bases on the strand of DNA, which will then be able to form bonds. So they'll be able to bind to complementary bases on other strands of DNA, which will be the plasmid, as we'll see further down in the question. 4. Explain how enzyme 1 in figure 1.1 is specific to the exact sequence of DNA bases. So enzymes are specific to their substrates in that the shape of the active sites of the enzymes are complementary to their substrates. So similarly over here, the active site of the enzyme in figure 1.1 is going to be specific to the DNA base sequences as shown in the figure. C. Another enzyme, enzyme 2, is used later in the process of genetic engineering. Figure 1.2 is a diagram showing the action of enzyme 2. So you've got the plasmid DNA strands and the DNA molecule, which you have just cut using the enzyme 1, which was a restriction enzyme. And enzyme 2 is going to combine the two DNA molecules together. And this enzyme will be DNA ligase. Since DNA ligase is responsible for adding the DNA, which contains the desired gene, into the plasmid. 1. Some organisms naturally contain DNA in the form of a plasmid. State the name of the type of organism that naturally contains plasmid. Those are bacteria. 2. State the name of the enzyme 2 in step 7 of figure 1.2. Again, that is DNA ligase, since it will add the DNA which has been cut out into plasmid vector. 3. State the name of the molecule formed in step 8. So this is recombinant DNA, since it contains DNA from two different species. D. Sketch a graph to describe how the activity of the enzymes used in genetic engineering would change if the reaction occurred at a range of temperatures from very cold to very hot. Label the axis with appropriate titles and do not use units or number scale. Here I've drawn the graph and labeled the x-axis as temperature, the y-axis as enzyme activity. Alright, so everything that I've drawn with blue is the only answer, and the black labels are just there so I can use them to explain the answer. You don't need to use any of that. So the region A starts from the origin and up until this peak point. During this point, as the temperature increases, the enzyme activity will increase. As the molecules will gain kinetic energy, the collisions will, the rate of collisions between enzyme and substrates will increase. At point B, you've got the optimum temperature where the enzyme activity is the highest, and beyond this point in region C, as the temperature increases, the enzyme will denature, the shape of the active site will change, and so the activity of enzyme will decline. 2. Figure 2.1 is a photomicrograph of the end of a plant root. Figure 2.2 shows the results of a study on the rate of uptake of nitrate ions at different points along the root shown in Figure 2.1. So the rate of the uptake of nitrate ions, just like other ions from the soil, takes place in the root hair cells, which you can see are present from 10 millimeter lengths. From the length of 10 millimeter up until here somewhere around 27 millimeter, and this is represented in the graph. As the distance from the root tip increases, the rate of nitrate ion uptake will increase, because as the distance from the root tip will increase, the root hair cells will become more common, and then above a certain length, which is somewhere around here, the root hair cells will not be present any longer, which is why you will see this decline in the rate of uptake of nitrate ions. A1. Describe the rate of uptake of nitrate ions along the root. Use the information in Figure 2.1 and Figure 2.2 in your answer. This question is of 5 marks. So I'll describe how the rate is going to increase and then decrease. This is the general trend, and the distinguishing point over here is that the rate remains constant between 10 to 15 millimeters. And as I mentioned earlier, as the number of root hair cells will increase, the rate of uptake of the ions will increase, since that is where the ions move from the soil into the roots. And since this peak value of the rate occurs at 27 millimeters, that means 27 millimeters is where most of the root hair cells are present. Before 2.5 millimeters of distance from the root tip. To explain how nitrate ions move from the soil into roots, so ions move into the root hair cells by active transport. This will require energy, as ions will move against the concentration gradient using carrier proteins in membranes, as ions are present in a larger quantity in the soil than in the roots, which is why they are going to use active transport for their movement into the roots. 3. Explain why the uptake of ions such as nitrate is important for the uptake of water into roots. So as ions will move into the roots, the water potential will decrease. This will allow water to move down the water potential gradient from the soil into the roots by osmosis. 4. Explain why plants need nitrate ions other than for the uptake of water. The nitrate ions are required to make amino acids, which will be required for protein synthesis, as amino acids are the building blocks of proteins. These proteins can then act as carrier proteins in the cell surface membrane. So here, as you've got 3 marks, you'll get 1 for mentioning a function of proteins in the plants, which I've mentioned could be a carrier protein, which would be required for active transport. 5. Describe how nitrate ions are formed in the soil. So proteins in the waste of animals that is laid on top of the soil is broken down to amino acids, and amino acids are then converted to ammonium ions by decomposers like fungi and bacteria. The ammonium ions will then be converted to nitrate ions by nitrifying bacteria. Other than this, nitrogen-fixing bacteria in the root nodules can also convert nitrogen gas from the air spaces in the soil to ammonium ions and other ions like nitrate ions in the soil. B. Figure 2.3 shows some of the events that occur when high concentrations of nitrate ions flow into lakes. Put the events in Figure 2.3 into the correct sequence. Alright, so first off, you've got an increase in the population of producers. This occurs when nitrate ions are leached out of the soil and they move into the water bodies, which will cause an increase in the growth of algae and green plants. So they are producers. They'll increase in numbers. This will be followed by a decrease in light intensity at the bottom of the lake, since these producers will cover the top of the lake. And as a result of this, a decrease in the population of producers will occur, since plants underneath the surface will not be able to get sunlight for photosynthesis, and so they'll die. This will be followed by an increase in the population of decomposers. So bacteria and fungi will grow, using the dead producers, and this will as they'll use up the oxygen dissolved in the water, it will cause a decrease in the concentration of dissolved oxygen, and as a result of this, a decrease in the population of consumers occurs as the consumers are not able to get enough oxygen for aerobic respiration, and so they'll die. To state the name of the process summarized in figure 2.3, this is eutrophication. Three, a researcher investigated genetic variation in fruit flies, Drosophila melanogaster. The bodies of fruit flies can be black or yellow. A yellow body color is a recessive feature in fruit flies. Two heterozygous fruit flies were black bodies were bred together. Predict the phenotypes of the offsprings and the phenotypic ratio for this cross. Here I've used the capital B letter to represent the allele for black color since it's the dominant trait, and small b to represent the recessive feature, which is the yellow body color. And so when you cross two heterozygous individuals, you'll have the two different alleles, one dominant, one recessive, and as you can see over here in the Punnett square, you'll have three of the individuals with the dominant trait, which is the black body color, since they have at least one dominant allele, and only one offspring will have the yellow body color, with two of the recessive alleles. B. In a fruit fly breeding experiment, researchers counted 124 offspring with black bodies and 121 offspring with yellow bodies. Draw a genetic diagram to explain the results of this cross. Use the letter B to represent the allele for black body color and the letter B to represent the allele for yellow body color. Alright, so in part B, you've got 124 black offsprings and 124 yellow offsprings. So yellow offsprings again have the recessive trait, which is almost half as many of the offsprings. So to have half of the offsprings carry the recessive trait, you'd need to have one parent being heterozygous and the other one being homozygous for the recessive trait. And so here you've got the genotypes and the phenotypes with the gametes produced from each of the alleles, each of the two alleles of the parents. So here I've drawn the Punnett square for this test cross, and half of the individuals will be heterozygous, whereas the other half will be homozygous for the recessive trait. And here I have given their genotypes, and the expected ratio is 1:1 since there are equal number of offspring of each genotype. C. The crab Cerberusa cica lives in dark caves and has no colored pigment. Figure 3.1 is its photomacrograph. 1. C. cica and D. melanogaster are both arthropods. So this one and the fruit fly that we are talking about. State one feature present in all arthropods but not present in vertebrates. That would be an exoskeleton. 2. C. cica is a crustacean and D. melanogaster is an insect. State one morphological feature of C. cica that distinguishes it as a crustacean and not as an insect. So it's got 4 pairs of legs over here, and insects do not have more than 3 pairs of legs. D. The ancestors of C. cica had pigmented bodies. The lack of a colored pigment in C. cica is called albinism and was caused by mutation many thousands of years ago. 1. Explain the mechanism that has resulted in the allele of albinism becoming common in the recent generations in populations of C. cica. So if a particular phenotype becomes more common in a population over many generations, it is because of natural selection. So natural selection will favor the survival of the organisms that are better adapted to their environment. And so here the albino crabs are better adapted to their environment, which makes them more likely to survive and reproduce. As a result of this, they'll be able to pass the allele for albinism to their offsprings, and over time and many generations, the frequency of mutant allele will increase. To state two factors that can cause mutations. Those could be radiation and carcinogens. 4. HIV is a pathogen that can cause AIDS. A. Describe how HIV is transmitted from one person to another. This can occur due to exchange of infected body fluids, which can be carried out by sharing needles that are contaminated with HIV, or blood transfusions without screening for HIV. It can also be carried out by breastfeeding. B. All viruses contain genetic material. HIV contains genetic material called RNA. State one feature common to all viruses. That would be the protein coat which surrounds this genetic material. C1. Describe the function of lymphocytes. So lymphocytes are responsible for producing antibodies, which occurs when the immune system is carrying out an immune response. The lymphocytes will produce antibodies and they'll also form memory cells. These memory cells will remain in the body, so they'll provide active immunity, which is long-term immunity, so that when the body is infected for the second time by the same pathogen, the person will not have to suffer these symptoms and they'll be able to recover faster. To state how infection with HIV affects the lymphocytes if untreated. So HIV, this virus can decrease the number of lymphocytes in the body, which makes the immune system of the infected person very weak. The doctors wanted to determine whether dietary supplements could help people infected with HIV. They randomly put volunteers with HIV into two groups: a treatment group who received HIV medication as well as additional vitamin and mineral supplements, and the second control group who received HIV medication but no additional supplements. The details of the two groups are outlined in Table 4.1. The dietary supplements were given to the treatment group twice a day for 3 months. The nutrients in the supplements included vitamin C, vitamin D, calcium, iron, other minerals, and vitamins. 1. Explain why vitamin C and iron are important in the human diet. So vitamin C is required to repair tissues and to make the collagen protein, which is present in nails, hair, and skin. Iron is used to make hemoglobin, which is needed for oxygen transport. Table 4.2 shows some of the results from the study. To use the data from treatment group shown in Table 4.2 to calculate the percentage decrease in the average number of copies of HIV RNA per centimeter cube of blood, and you're given the space for working. So to find out the percentage decrease in the treatment group, you've got the treatment group data here, and a decrease in the number of HIV RNA. We would find the change, that would be 4291 minus 897 divided by the initial value, which is this, and then multiply the fraction by 100, and that gives the answer as 79. To evaluate the effect of the dietary supplements on the lymphocytes, use the information in Table 4.2 in your answer. As you can see in the treatment group, for the people who were given dietary supplements, the number of lymphocytes is going to increase after 3 months. So after they've had the dietary supplements, they are facing an increase in the number of lymphocytes. In the control group, who were not given any dietary supplements, the number of lymphocytes remains the same. To use the data as mentioned in the question, you can write the exact numbers. So you can write about how at the start of the treatment the number of lymphocytes was 357, and after 3 months it was 422 cells per micrograms of blood in the treatment group, whereas it remained at the same value of 461 cells per microgram of blood in control group. Figure 5.1 shows fishing on a large scale and a small scale. All types of fishing can have a negative impact on fish stocks. A. Discuss how governments can regulate fishing to maintain fish stocks. Use the word sustainable in your answer. To maintain fish stocks, you want their numbers to not decrease, and this can be done by using sustainable resources. A sustainable resource is a resource which is produced and removed from the environment at the same rate so that it does not run out. And for this, governments will need to monitor fish population, limit the fishing season so that in the season that the fish breed, fishing them will not be allowed. Set down quotas for fishing, raise awareness for the decrease in the diversity of fish to those who consume it, and arrange for protected areas of the sea where fishing is prohibited to protect endangered species. Here I've got the spelling wrong. B figure 5.2 shows Figure 5.2 shows the location of a chemical factory near a river. Figure 5.3 shows the sex ratio of the fish Catostomus ecomersoni in the river at site 1 and at site 2. So the direction of river flow is from site 1 to site 2, which means that anything which is excreted from this factory will not be present at site 1, but it will be present at site 2. So the effects of the waste material of the factory will be seen or observed at site 2, as water at site 1 is not contaminated by the waste. Intersex fish have both female and male reproductive organs. State and explain what type of chemical the factory could be releasing into the river that would cause the effects shown in figure 5.3. So as you can see, at site 1 the male to female ratio is close to 50, but at site 2 there are very few male fish compared to a much greater portion of female fish, and you've also got many more intersex fish, which means that the chemical factory is secreting female hormones like estrogen and progesterone, as at site 2 the number of females is much greater and the number of male fish is very less compared to site 1. C. State how sex is inherited in humans. It's inherited in the form of sex chromosomes that are the X and Y chromosomes. Six, the heart pumps blood around the body. A. Explain why the heart is an organ. So an organ is a group of tissues that are working together to perform the same function. B. Complete the sentences. The circulatory system includes the heart and blood vessels. The oxygenated blood from the body is transported to the heart in the vena cava. During a heartbeat, the ventricles contract. The right ventricle pumps deoxygenated blood into the lungs. The right ventricle has a thinner muscular wall than the left ventricle, and that's because the right ventricle only has to pump blood to the lungs, which is a shorter distance from the heart compared to the entire body, as the left ventricle has to pump blood to the entire body, so it's going to have a thicker wall. Gas exchange in the lungs occurs by diffusion, as gases will diffuse from the blood capillaries into the alveoli and the other way around. Oxygenated blood travels back to the heart where it enters the left atrium of the heart. Two sides of the heart are separated by septum. This structure prevents the mixing of oxygenated and deoxygenated blood. Oxygenated blood is then delivered to the rest of the body. Blood is supplied to the muscle of the heart and the coronary artery. Now, this supply is to the muscle of the heart and not to any of the chambers like the atrium or the ventricles. So keep in mind that vessel is going to be coronary artery. C. Many people monitor their heart rate by counting their pulse. State one other method of monitoring heart rate. This could be done by ECG. Alright, this is it. We are done with this paper. Thank you for watching.